what you're doing. Just to make sure, can I get like a thumbs up if you could see the screen? Are we good? Okay, great. Okay, so today I'm going to talk about data integration, and I'm going to talk about that generally, but then also thinking about that in the context of macrosystems biology. So, what I want to do, my goal today is maybe to demystify some of what is data integration and talk about why specifically it's useful in macrosystems, some of the open challenges, and then I want to end with like some tips on getting started and thinking about where to go from here. So that's kind of the progression today. All right, so let's just dive right into it. What is data integration? Okay, so first of all, I want to say that it goes by a lot of different names. So you may have heard different comments for this: data assimilation, integrated models. If you do anything with populations and communities, you could hear about integrated population models or distribution models. But basically, the idea is the incorporation of multiple different data types on a single system into a unified modeling framework. So that's where we're modeling different data types on whatever the target is. For example, an organism, not adding in other data like covariance. So if you're adding in climate data or weather data or something like that as an influence on something, that's not the same as data integration. Data integration is where we have multiple data sources on whatever it is the thing that we specifically want to study. So typically, like there's a lot of scenarios where data would be collected, but then they, like on a target organism, let's say you're looking at a specific bird, but then they're analyzed kind of separately. And so the concept of integrated analysis is kind of developed in fisheries in the early 1980s. And since there's been a variety of approaches where the basic idea is we want to model all those data together instead of doing a separate by separate analysis and then reporting those results. All right, so where do we go from there? How does this work? So the basic idea when I think about data integration is I kind of start, I mean, it's a little bit different than when you're thinking about starting with your question. We do obviously want to think about that, but one of the ways I think about it for the actual like analysis is I think about what are the data types, what kind of data do we have available? And there's a variety of different data that can be used. So here my examples are on, a single species, and you might have some information on, where it's been seen across its distribution. You might have count data. You might have something about reproduction or survival if you're capturing individuals through time. So that's kind of where one of the first ways you want to start with this is thinking about, all right, what are the data available? And then I would say with that data available in mind, you want to specify whatever is the biological process that you want to model. So in an ideal world, with this thinking about population is you might want to really try and model that in a way where you're explicitly estimating things like survival, reproduction, looking at the processes, birth, deaths, immigration, that type of thing, to look at the processes that are actually influencing whatever it is that state variable that you're interested in. So maybe abundance through time. Now, obviously, that's the ideal, but that's not always the case. Sometimes we're kind of doing it a little bit more phenological, like where we're not really modeling the process. We're just thinking about, this is on the left here, we've got the breeding or the distribution of the cerulean warbler. And maybe what we're actually trying to do is in Michigan, model something related to how the forest cover is influencing the occurrence of cerulean warblers in the summer. So in this model, like the structure is not so much, modeling that full, like, detailed biological process, but just something related to the counts, something like that. We can do that in both ways. And I would say, I'm happy to discuss this more when we go into the breakout rooms, but that biological process model, how detailed you're going to be able to get with it, is going to be at least somewhat driven by the data are available. You might want to be able to model every single process about how the cerulean warbler survives and everything. But if the data that you have is just the occurrence locations or maybe just simply the abundance, it might not be realistic. So those things, developing that process model, the biological process model, is going to be related to those data that are available. Okay, so once you've determined that, the key thing is to figure out then how each of the data sources are informing the specific parameters. So here I have these blue diamonds, and each of them represents a specific data source. And so that's not really important what they are, but what you're going to do is kind of conceptually think about each of your parameters. So in that first one, when we did that life cycle, for example, let's say SJ and SA, survival of juveniles, survival of adults, you're going to think about what data sources provide information on that. And that might be something like the capture and capture. Hey, we're capturing individuals through time and measuring survival. But even something as simple as count data through time does contain that information. So the key is then we're thinking about, all right, which data sets that we have available are informing the parameters of our biological model. And so we specify the model that way. Really, I mean, with a pen and paper, I'm writing this down. And then on top of that, the final step is I think about with these green boxes, these green triangles, is how is the error or something about the detection or sampling process influencing those data that we have? So it's, if, for example, if you're looking at count data, right, you're not seeing the true counts of everything. There's some error around that. If you have mark recapture data, right, you don't necessarily detect every individual each time. So the final process is to think about what's influencing my sampling process. And the nice thing about this is you don't really have to do this all by yourself, because each of these individual data sources have already a huge literature on how people go about all of these different processes. So it's not like if you're doing this, you're going to be starting from zero. Okay, so let's just go into why. Why would you want to do data integration? So the biggest one, and the main reason everybody's doing this, is it increase accuracy and precision in biological estimates. So here's an example from one of my student Matt Farr's work. Let's say you have an ecological predictor on the landscape. So that's the thing on the left. And what you want to do is estimate abundance and the effect of the landscape. So if you just had something like presence only data, that's just like out. You're only saying, all right, it's here, nothing about where it's not, anything like that. You can see that you can get an effect of the landscape. You can estimate that, but actually your estimate of abundance is unidentifiable. And that's why you see that box there on the top abundance line is just nowhere close to the true value. It's actually just unidentifiable. But if we add in a little bit of this structured data, in this case, we're thinking about distance sampling data. It's not important what kind it is, but you can see pretty quickly that not only does the accuracy improve, even with a tiny percentage of that, but then as you increase that amount of data, you get a really high, better precision. So you get more accurate, more precise estimates of whatever the biological process is, in this case, abundance and the effect of a covariate on abundance. All right, so another thing is that we want to understand the factors influencing population dynamics. So here's an example with black terns. So there was a population of black terns. This actually data comes from Michigan, where the U.S. Fish and Wildlife Service were collecting information on the black terns. They're doing abundance, capture, productivity data, and then they saw that this population is declining. So there was real worry about that. And what we were able to do by integrating all those different data sources together is we were able to figure out that it was actually adult survival. So that's the bottom right corner that was really driving that population size. So we're able to figure out what of the demography is causing that decline. In this case, it wasn't something like fecundity or, something related to reproduction or juvenile survival. It was really those adults surviving. And then what we did is we think about, all right, we can use this data integration to get a better understanding of the prediction of the future and future vulnerabilities. So this was, what if we looked at extinction probabilities into the future based on if we do no different management, so that's the yellow dashes there, under different climate scenarios, so that's with different things related to the NAO index, so like what was happening with climate. And then what happens if we increase fecundity, juvenile survival, adult survival, and what you can see, so that's the dark blue lines, is that increasing adult survival is kind of the only thing that really makes, except for obviously doing everything together. But if you're going to do one thing, the thing is, if we increase adult survival, we can really reduce the quasi extinction probability fairly quickly, even within 10 years out from no from starting management. I just said that. So here's the increased survival. The adult survival is the main thing you want to do. All right, so now let's take it back and say why data integration for Macrosystems. Well, I know this group all knows what Macrosystems is, but just in case there's any new students here, Macrosystems, ecology or science is the study of ecological patterns and processes at broad scales, but also their interaction with the phenomena at other scales. And I think that's also sort of the important part. It's like we want to understand how what's going on at a large scale or a small scale is influencing those things in between. And so one of the reasons data integration is really helpful for macrosystems ecology is because of the types of data that are available. So at a broad spatial scale, so at across a continent, we tend to think of the data that are available as being something like unstructured with lower information content. So something like any kind of citizen science, participatory science. If you think of something like iNaturalist, you've got that at a really, really large spatial scale, right? Across the country, maybe. But each of those specific, data points where someone's uploading their iNaturalist observation doesn't contain that much information by itself. And that's partially because it's just unstructured. It's not collected with any kind of sampling design. And on the flip side, at small spatial scales, we can go out and we can collect really high information content that structured data, so data that comes from a study design, but those tend to be spatially and temporally limited. So exactly, if we go back to thinking about the capture recapture example, it's really difficult to collect recaptures on a really broad spatial scale. Typically, we're doing that at like a breeding site at a small scale and focusing just on that because it's just in practice not realistic to have people collecting those information, those data across a really large scale. Okay, so one of the things that my lab has worked on for a really long time is understanding butterfly declines and specifically the monarch butterfly decline. And I think that this is a great case study for thinking about data integration and really thinking about it at a macro systems. And the reason for that is a few things. First, and I want to go a little bit into this case study to give you an example of like, okay, how did we go and what are the types of results that we see in this? So in this case, the time series suggests multiple imposing factors on population dynamics. Because they're iconic species, there's a lot of potential data sources, and agencies are recognizing that the species is in decline, and particularly the migratory individuals. So I just want to acknowledge the team that has worked on this on this project I'm going to talk to you, because one of the things I think that's true about data integration is, and especially at the macro systems, is you end up needing a lot of people who have different expertise, right? Some people related to those data modeling, on the ground, biology, all different things. And I think that's even more true with data integration at a macro scale. All right, so first, monarch butterflies are declining. This graph here shows the area occupied by monarchs in colonies in Mexico. So that's where monarchs spend their winter. And that's the best way to kind of delineate the size of the population because the rest of the time they're spread all over the continent. And so the monarchs are spending their winters together in a few different colony locations in Mexico, central Mexico. All right, so to understand what's going on, why this is a macro systems problem, you got to understand a little bit about monarchs. And I'm going to say this different than the pace of this slide because I always mess it up. So monarchs overwinter in the high elevation Oyamel fir forests in central Mexico. And then in the spring, so about early March, they kind of wake up, they reproduce, they copulate, and then they fly up to like Texas and the surrounding area, and they produce that first generation in that spring migration breeding period. So Texas and East Texas. And then they produce 2 to 3 more generations by expanding further north, their milkweed, their host plant is milkweed, and they're obligate milkweed feeders. So basically, they have to lay their eggs on milkweed, and the caterpillars enter different instar stages and are eating that milkweed before they become butterflies. And they spread out to all of eastern North America. So in this case, I'm really talking about this migratory population. There is a population west of the Rocky Mountains, but they spend a different place overwintering. So we're talking about everything east of the Rocky Mountains. And then finally, in that last generation, at the end of the summer, early fall, they go into what's called reproductive diapause, and they fly all the way back down to those same sites in Mexico. So multi-generation migration back to the same sites. So one thing that you can see maybe right away is that the dynamics from one stage carry over into other stages. So one thing that's important is, the Eastern US population, it's all one population. Everything east of the Rockies, there's no genetic differences. They mix, they breed together. And so they're really all one population. And it's the details of this figure aren't important, but basically what we're showing is what the counts of monarch butterflies are. The number of butterflies seen in Ohio is related to the spring precipitation and spring temperature in Texas. So really just showing that the effects from one stage really carry over to another, and that because of that, it's difficult to tease apart the effects of local or regional influences. So actually, this was my first Macrosystems biology grant from NSF, and really this kind of case study is what brought me into Macrosystems. So there's been a bunch of different hypotheses for why monarchs are declining, but there's three main ones, and those I'm going to go through each of these to tell you a little bit about that. So one is the milkweed limitation hypothesis, and this is the idea that the adoption of herbicide-resistant crops led to lower milkweed densities in agricultural fields. So on the top here, I'm showing you the size of the early wintering population with this dashed line around 2004, 2005. And what you can see is that before that time, on the bottom there, the bottom is the amount, the proportion of glyphosate use in a county. So like the proportion of crops sprayed. And so what you can see is a really rapid increase in the Roundup, basically from the early 90s to the mid 2000s, about this 2004 time period. And after that, we see a kind of stabilization, but at a very high level. So across the Midwest, there's a lot of this herbicide being sprayed. Okay, so then another one is this hypothesis on, it's called the migration survival. And the idea for this is that it's actually not the summer population. It's really the effect of the migration related to reduced nectar availability, disease, loss of forest habitat, and that's led to lower survival. And the reason for this hypothesis is really that top graph. So that top graph shows the expected summer count at one of the biggest sort of most broadly collected data participatory science sites. So NABA is the North American Butterfly Association. And really, if you just look at like what are the abundance, the counts at those locations through time, you don't see that same decline as we see in that winter population. So people were saying there's a mismatch between that. And so maybe that's the issue. And then there's also been evidence that the amount of greenness on the landscape and the amount of forest cover available at those overwintering sites does really influence the size of the overwintering colony. And then the last one is the climate change hypothesis. And that is that the direct and indirect effects of climate change have led to lower fecundity and survival. And because of that, we just see fewer monarchs. And there's evidence for this, in the lab. So, and in other places where it's like, monarchs, like all insects, they need really specific climate conditions. And if we don't see that, what we're likely to see is, not success. So there's laboratory evidence for this and some field evidence as well. So our question was really, what is the relative contribution of stressors along the migratory route to monarch declines? And the idea for that is we want to develop a full integrated, full annual cycle model. And this work was mostly completed by my former postdoc, Aaron Zylastra, who led the development of this. And the idea is that we wanted to use data from the winter, late winter, monarch data from the late winter, from the summer across the different breeding ranges, and the early winter to develop this annual cycle model. And one thing I'm going to note here is we were really only able to do this from that year 2004 onward. So this is after the time of that glyphosate spraying has kind of leveled out. And past the time of the steepest declines. And that was just because we really tried to go back to the 90s, but the data just weren't available. That was the original goal, but it wasn't something we were able to do. All right, so we broke this down into two subcomponents, a late winter to summer model, and then a summer to early winter model. So I want to just go through a little bit, how did we think about this? All right, so first is the summer submodel. So we first, going back to how do we do this data integration, we thought about what data are available. And there's really 5 different data sets that are available and 2 different types. So one is those census surveys I alluded to in the North American Butterfly Association. And that's where participatory scientists go out in a single day in a 25 kilometer radius, so really big radius. They're trying to do this census, count every butterfly that they see. And these are groups of individuals. So those are all these blue spots on the map. And what you can see that's a real advantage of that is that they're spatially widely distributed. So on the flip side are these state butterfly monitoring networks. And there's one in Illinois, Ohio, Michigan, and Iowa. And these are really different surveys. So in this, the individuals volunteer every single week. They walk the same transect, the same location. These are called polar walks, and they record the number of butterflies, each species and how many they see within a fixed meter radius or a fixed, transect on each side. So these are really different types of data. You could see what's good about those data is, right, we've got this repeated time during the summer as opposed to just one time in the summer from the NABA data, but you could see right away, right, they're spatially aggregated. They're only in those states. So we're missing them, for example, Wisconsin, Minnesota, and those are important parts of the monarch range. Those are considered to be really important parts. So the first thing we had to do was add them together, figure out how to put them together and model this. So one of the things that you're going to notice, I'm zooming in on the area, and this is the area outside of Chicago. So that's Lake Michigan there. That's not really identified. And then there's a bunch of spots. So we're going to get back to this. But one of the problems, right, with participatory science data is that they're aggregated where people are. And so that makes things really difficult. We have to think about how do we model that. So what we decided to do was we model each survey location. So there's an example, a little red one. And we say that this Y, the data that we're going to see, that's the Y, is proportional to, is related to whatever the mean count is for the county. And then something related to covariates on the survey methods. So which different type of data was it? And each of the different actually surveys in the different states have slightly different protocols too. How much effort was done, how much time was spent. Luckily, we have that information. And so we incorporate those into what we think of the, as the data we're seeing. So the data that we're seeing is going to be a function of what the actual mean count is, and then what's related to the sampling methodology and effort. And then the next thing is we're going to say, all right, we actually want to model what the mean count at a county level is related to the ecological covariates. So here we included something about what's the size of the late population in winter, what was the spring weather variables look like, precipitation, growing degree day, all that sort of stuff in Texas on the way up. And then what does the cumulative temperature look like, in the specific location in this county? How has weather been? And is this location, does this county have a lot of crops, and how much herbicide are they spraying? So we're able to do that kind of at the county level by accounting for all those differences. The other nice thing about that is, right, in this county where we're highlighting, there's 3 surveys, but in some county, there might be, many more than that. We don't want to overcount those. All right, so then the next is this winter submodel. And the way that this works is we have these individual colonies, that where we have the proportion of the area occupied by monarchs. So essentially what's happening is we got these colony locations, and you can see the purple line around them kind of is the larger colonies. So there's little spots within them, but largely we're saying on the tips of these mountaintops, how much area is occupied. And so there's not really just one colony, but actually a few together. And we say that that area occupied, conditional on it being used that year, is related to the ecological variables. So again, this condition on being used. So you can see in this chart on the left, right? So this is the size of the various different colonies. Sometimes the counts for some or even most of those colonies, the smaller ones for sure, are 0. So we need to account for that in some way. And then we want to have ecological covariates related to what is the summer population size, how much nectar was available during the migration down, and what was the forest cover in early winter. And then what we do is we need to connect this summer component of the model to that winter component of the model, to the early winter. And so what we do is we develop this index. We go across that full county in the Midwestern range, across the entire area, and we take and a weighted average by how much land is available in each county for butterflies, so unforested land, over that breeding range to get an index value for the year. So like how big was that population at the end of the summer? How big is that population of individuals right before they're about to do their migration? And then we use that index for as that peak summer index in the winter component of the model. And that's how we put them together. So what did we find? Interesting thing is we found strong support for the climate change hypothesis. So here's a metric that shows the proportion of each of the different variables on explaining the relative importance. So we saw strong effect for both weather in the spring and the summer. So spring and summer weather conditions were more than 7 times more important than other factors explaining variation. And part of that could be related to this thing that like 74% of that variation in glyphosate use, so the Roundup use, was attributed to differences among the counties, whereas there was only, less than 26% of the variation was temporal. So that might mean because we're seeing, we're past this saturation point. This tells us nothing about what happened from the mid 90s to the mid 2000s, but post, 2000, 2002, 2004, we're seeing that this spraying of the crops doesn't appear to have the biggest effect. And then what we found is that the size of the summer population was explained more than 90% of the variation in the winter population size. So a really, really tight link. And, what was interesting about this is that here's our monarch count, our summer adults count. And we actually went back in time and did this for the time from all the way back to 1994. But what you can see is that once we accounted for all the different data, like if we didn't just take the peak index from one survey type from the NABA, the North American Butterfly Association, we actually did see that variation in the population and the size going down. So maybe not surprising. And then what did we find? That temperature increases can increase monarch abundance, but not in the warmest areas. So this shows like deviations. The x-axis is the deviations from the norm in growing degree days. So like on the right is hotter, on the left is cooler. And if you look at different sites, like what is the count for, monarch abundance based on if you're in a hot location. So that top one is the coolest location. So that would be like in Minnesota. And you can see that as you get hotter, you get more monarch abundance, but that starts to decrease when you're in those warmer locations, which is that like those dotted lines. And then what happened is, unfortunately, during this specific time period, the difference between 94 to 2003, from 2004 to 2018, is that actually those northern Minnesota places or whatever, those places where we would want them to be a little hotter, they were cooler, and it was hotter actually than average in those sort of the southern sites. So just to note, this isn't what the prediction for the future is like. These areas are predicted to get hotter, but that's what actually happened. We actually saw that during those decades that we were looking at, some sites were a little bit cooler. So one of the things we talked about with this data integration is this is a helpful approach for predicting into the future. So one of the things we wanted to do is say, all right, what would the future look like for monarchs in these locations? And so this on the left is the average of like the counts. So you can see these Minnesota maybe have a little bit more, some of the northern parts, the expected counts. And then what might that look under different scenarios of low emissions and high emissions? And if we look in the near term, under a low emission scenario, actually, there might be some places, interestingly, that the southern area where you're not expected to get quite as warm. So those northern areas were predicted to get really, really warm more quickly. And you have this east-west gradient of precipitation. So in these areas like northern Ohio, where it says maybe monarchs might do a little bit better there in the future, which is not generally the case that you think of as producing the most monarchs. That's more in the upper Midwest, but it's predicted to get wetter and not quite as hot. It's getting hotter, but not as fast as some of the other more northern places. But I would just make a comment that under a higher emission scenario and longer timeframes, right, we're going to expect to see declines across the Midwest for monarchs. Okay, so that's kind of wraps up what we were doing with monarchs. And the point with that was just to try and show, all right, how do we put them together? What kinds of questions that we can ask? And before we end this, I just want to briefly talk about some of the challenges with data integration across scales. And I had heard that, Lori mentioned that some of you were maybe looking at a paper that we had done previously addressing data integration challenges across scale from 2021. And that was a special issue in Macrosystems. And, all I'm just kind of showing here with this paper is that, the number of studies in both Macrosystems and data integration are growing. So people are, going to be using these methods more and more. So one of the big challenges, so I wanted to kind of address some of these challenges and think about where are we in this state. So one of the open challenges in data integration is resolving spatiotemporal mismatches in available data. And this is especially true at Macrosystems, right? So on the left here, we've got some kind of remote sensing satellite, and maybe this is happening at a really large scale. And then you can think of something in situ where you're collecting, water sample at a really, really fine scale. So you can see right away that one of the problems is we got to figure out how do we put those things together. And I'm just going to go with a little example that shows, okay, what's the problem if we don't do this? So this is some work by Krishna Pacifica, and they were looking at black-throated blue warbler across Pennsylvania. So they used the breeding bird atlas, that's the BBA, and eBird, and those are collected on very different spatial scales and aggregated at different spatial scales. And so if you just naively kind of put this together, it shows these kind of big hotspots, but those hotspots are more related around to where the data collection is happening. And then they use this approach called change of support, where you could see kind of a much more realistic distribution of the species. So really, change of support is just a process by which you're either upscaling or downscaling the data to achieve a single extent. And usually it's the upscale because that tends to be easier to do. And it's different than interpolation and regridding that are common in the GIS softwares or something like that, because it takes into account uncertainties associated with changing scales. So this helps the change of support preserves that information content in each of those. So the nice thing is this is a good approach, that people can use. And there's a lot of literature in the statistics literature about how to apply change of support. And there are some sort of very straightforward ways to do that. Another is unbalanced data. So you can think of the example I have here is kind of an automated machine where you're collecting, data every minute or even every second. Whereas if you have someone going out into the field and collecting data, you're getting that at a much lower resolution, right? You can't be doing that all the time. So some approaches to that with this unbalanced data, a lot of one type and then a little of another, you can just, maybe subsample and downweight for larger data sets, but that can also lead to subjective choices. So if you do that approach, it's not a bad approach necessarily, but you want to at least think through that and see how sensitive your results are to that. And then there's active areas of research that show, how you can weigh, you might be able to weigh unstructured data. So like if it's participatory science or that other kinds of data related to something like the observer expertise, you can model those factors that inflate that information context. So you can use things like autocorrelation or formally put that directly into your model. And this is just an active area. There's no real consensus on how to do that, but there are a lot of different ways out there on how that might be done. Okay, accounting for biases in data sets. So we talked about that with the example that we showed, right? This is especially true with participatory science data, where we're not necessarily having a design. We tend to see that data are collected where people live, which is not necessarily surprising. So there's a huge range of solutions depending on the specific of the problem and how you think that it will be manifest in your question. But one of the things I think is the biggest is just explore the data independently and together to identify the problems. Like, can you see that with your individual data? And you could use model components in there to describe that site selection. Or a big one is people use random effects in their model to model those processes in there. So again, no real right way, but a lot of different possibilities. And then another one, and this is a really one non-stationarity that affects macrosystems research in particular. So I think that this challenge of data integration is really the non-stationarity is a big one for macrosystems research and maybe not quite as much for small scale. So the idea of non-stationarity is that your effect of your parameters are changing through space or time. So like a stationary variable, what we got on the left here shows a similar trend over the entire spatial range, right? So the left is the spatial range or a temporal range. And you just see the same, effect. There's nothing going on. But in some scenarios, what we call first order non-stationary is that a change in the mean of the effect of some parameters. So like, where in one location, this is like an east-west kind of thing. In one location, let's say you have, a positive effect of temperature, and another you might have a negative effect of temperature. That can happen for a variety of different reasons. And then the second order one is where the variance changes. So the effect of the parameter, maybe the means are the same, maybe the effect of precipitation is the same in both the west and the east, but there's more variation in one side and the other, and that also happens for a lot of different reasons. So solution kind of depends on the effect and magnitude of the non-stationary. And again, this is something you just have to think about at your large scale, like what that might be. And I would say another thing you can do is, start looking through your data, like, can we see that this might be happening? And there's ways to do this if you have, differences. If the means are just different, then you can incorporate some kinds of effects, right? Maybe you're just modeling it differently between the two locations or have some hierarchy in there. And then another thing you can do is you could actually index those parameters by space and time. So allow those covariate effects to change. But the problem with that is that always requires more data, right? So you might want to do that, but it might be hard to actually do. Okay, so we talked a little bit about what's the value of data integration in macro systems, and I showed a case study, and then also talked about some of the challenges. But I want to leave with a note of, all right, where do you go from here? How do you get started? How do you think about that? And one thing I think with data integration is that these projects start the same way as all the others, and I would say the number one is plotting your data, right? So unlike in data integration, I'm going to guess in most of the cases, if you're doing this data integration, these are not data that you're collecting by yourself. So mostly these are data sources that you're probably getting from somewhere else. Maybe you're incorporating some data that you've collected, but really in these macro systems data integration, it's not like we're designing a question, going out and collecting our data. It's a little bit opposite, right? So you're usually getting the data. And I think that the big thing that, sometimes I notice that people forget is, you just want to approach it the same way. Like, start plotting the data. Does it make sense? Can you see the trends? Can you figure out if there are outliers? All the usual stuff that we're doing, before you make any specific decisions about what's possible or what to do. And you might, obviously you've got some goal in mind of what you want to answer, question that you want to answer. But before you go too far down that, just see what your data are telling you what's possible. You'll get a pretty good understanding from that. And then I would say, Once you do start thinking about your question is start simple and build up complexity. I feel like I'm telling this to my students all the time, right? Because they're so excited and they want to go into the most complicated model to answer, but that is not going to work. It almost certainly will not work, right? So you want to start simply, like build your model. Can you just estimate abundance? No covariates. Can you get it to converge? Can you get it to make sense with your data? Add in one single covariate, then go from there. So I think, keep it simple. That'll be really helpful for troubleshooting because then you'll be able to figure out where your problem was. And then the other thing I would just argue is that, more complicated isn't necessarily better. I mean, you might have this idea, hey, I really want to do it in this hierarchical way and adding in this and that, but it might not change your inference. And so why make yourself so crazy, right? Like, if you have a reasonable justification for what you're doing, I think it's okay to move forward. And along those lines, right, I think this is really important. There is no right answer, right? So there's no right way to do this. There's no, if we knew the truth, we wouldn't have to build a model. So if something's not working or you're trying things or you have a way that you think your structure of your model, you want to do it this way because you think it meets the assumptions best, but it doesn't work. So then it's okay to revert to something that isn't exactly right. I think this is the big thing, all the time. I remember when I was first starting this and, I was developing these models. They were like these hierarchical community models, multi-species occupancy kind of thing. And, we were writing out all these assumptions of the models. And I remember the reviewers were like, yeah, those assumptions won't be met. Those assumptions. And I was like, this is literally the assumption of every t-test, every ANOVA, whatever. Like these are just basic model assumptions. And we were just writing it out. And that's because people don't think about that when they're using a t-test or an ANOVA, what the assumptions are. So you're like, I guess what I'm trying to say is people are violating assumptions all the times with their analyses. And the point is to figure out if that's a problem, right? It's not necessarily, it can be okay that you violate assumptions. I think we do that all the time. It's to figure out the problem. And what I always say is, we just think about the best way that we can do it. We publish our code. We try to be very, very, very transparent on what we do. Here's our model. Here's the structure. Here's the code. And then someone in the future can say, all right, that was dumb. I don't want to do it that way. But at least, we've put it out there and we've put out why we think it's out there and the best that we can do. And then someone else can just do it a different way when they think that what we did doesn't make any sense. And then finally, I think, just find collaborators with complementary expertise and skill sets, because I do really feel that data integration is hard. It's really hard at a macro system scale, and so there's going to be different people that have different expertise. And really for us bringing it together was the main thing that allowed it even to happen the way that we wanted, right? So it's like, I can do this and someone else can do that and we can put it all together. So that's all I really got for you. I just want to acknowledge some of the other people who contributed to the work that I presented here and our funders. And yeah, I'm happy to take any questions. I think I stopped sharing the screen, but I can put it back up if people do have questions.
Great. Thank you so much for that talk. That was excellent and very interesting. We are a little bit behind our schedule, but that's okay. We have some flexibility in terms of how long our report out period is and such. So I would like to see if maybe we have time, let's say for 2 or 3 questions as a full group. And then I would like to go to the breakouts so people have time to discuss more deeply. So are there any questions? Can people use the hand raise or anything? I'm looking on my screen, seeing if I see anything. Napaka, I see your hand up.
Hi, Elise. Thanks for the talk. The question is, if you're trying to work with, I guess this is a hashtag other people's data question. If you're trying to work with other people's data and you're not totally sure on all of the ways in which the methods that they used could create some of these biases that you need to make assumptions about and sort of model in, like, how do you handle the unknown unknowns, basically?
Yeah. I mean, you can only do the best you can do. I mean, I think this is where collaborators come in. I try to work with most people who I try to interact with people who know those data better than me. So usually, we'll have someone on the project. I mean, those, Leslie Reese, so is the expert on those NABA data and the state, and she's been working with each of the individual leaders from each state on this and different things about their protocols. So she knows that information better than me. So I would say one of the things is just trying to get people on your team who spend time, at least understanding what those protocols are. But you can only do as good as like what is written, what the metadata are for them. I mean, if you don't know that, there's nothing you can really do. And I'm fairly certain pretty much everything that I do, there's things going on. I mean, it's how like a modeler goes out in the field and you're like, oh, wait, what? That's how you're doing it. But I think the best you can do is just be transparent about it. So work with people who know it the best, and that, and then be transparent about what you did. And I think that's all you can do, right? If it's not written down what those protocols are, if you don't have those metadata, or I mean, hopefully you have the metadata, but if the information is included in the metadata, there's not a lot you can do.
Other questions? Maybe one more and then we can move to breakout. Emily.
Yeah, thanks so much for this talk. I'm really inspired by your we're just going to do the best we can do attitude. And I'm curious if, not having worked with integrating big data sets a lot, if there are certain assumptions you feel more comfortable or more confident making, and others that you feel like, oh, I'm not as confident about this. I'm thinking about the biases, and when you're trying to average across all these different places, are there ways that you can test to be like, oh, these assumptions really have a big impact on the predictions we're making, or these assumptions kind of get lost in the noise? And any insights you have about that, I think would be helpful for me, being very new to this.
Yes, I think you're exactly right. So the way you're thinking about it, you have to sort of figure out, is there something, are these assumptions going to hurt my inference? Sometimes you can't figure it out, especially if you don't know. But I think this goes back to the data, right? Like if you've plotted your data, right? Like if you've done those kind of checks at the beginning where you're like, yeah, okay, I see this positive thing. Butterflies. I should be seeing, they're not butterflies here in my model's not predicting that there are butterflies in December, like, so there are ways that you can go back, right? You can look and see, like, what is the thing predicting? What are the data saying? Are they reasonable to get from those things? Do those things make sense? Does that go together, right? If I'm doing a model and it's estimating these counts that are like unbelievable numbers not related to what we're seeing, okay, like that's a problem right there. So usually, I mean, that's the interesting thing I always say with my students too, is like, I can usually feel like I can diagnose what your problem is when I see the structure of what you've written out your model, and I've see what you've got for the results and what you've got for your raw data, right? And I think, that's kind of the same thing for every model and model type, right? You should be able to look and see, like, all right, there's no way this matches, right? And so then that means like some assumptions being violated, or your model isn't doing what you think it's going to be doing. So I think, again, just going back to the data, back to the data, um, and trying to figure out, yeah, does that work? Is that working? I mean, that's how we diagnose, like, oh, this distribution, like we used Poisson, and it's like, nope, that's not going to work. All the counts are way high. It's because it's an over dispersed distribution, but our predictions don't make any sense with our data. So I think that, yeah, does that answer it? Are there other things related to it?
Yeah, that's helpful. It helps me to get a little more sense of how one might get their bearings in the
Yeah, I mean, I think
sort of
you have to go back to what you have there, what makes sense with you. Expecting, take the average of your expected counts, do all this stuff. Like, are your predictions in that realm? It's kind of like the same way as the formal tests when you're looking at your residuals and things like that. You're kind of doing the same thing, but maybe in a slightly less formulaic way, because you won't have a function to be able to pull that out and do it with necessarily. Does that make sense?
Yeah, thanks.
Okay, thank you very much. I'm going to stop